Hello everyone, my name is Chedly Kastally. I will talk today about an ongoing project conducted in Finland on characterizing the genome-wide impact of tree improvement in Scots pine. First, let's talk about Scots pine. So what you see here is a map of the distribution of Scots pine represented in green. It's the most widely distributed conifer species, extending from southern Spain to northern Finland and ranging across Eurasia. Scots pine is an important species in Europe, both ecologically and economically. Several latitudinal clines have been measured in Scots pine. Here are two examples. You can see on the left the median number of days to bud site in greenhouse conditions, with the earlier stopping of growth in the north. On the right plot, you can see the survival rate in natural populations to height 2.5 meter in local environments, with higher survival in warm conditions. These data reflect the importance of local adaptation in Scots pine, which is taken into account in breeding programs by using several breeding zones. The main targets for Scots pine breeding within each breeding zone include increasing growth and wood production, improving timber quality, and improving resilience and stability across environments. It's also essential to maintain genetic variability to avoid inbreeding, and this is going to be the focus of this presentation. So breeding Scots pine can be summarized in the following steps represented in this diagram. First, from natural populations, trees of high quality called plus trees are selected based on their phenotypes. These were tested with open pollinated progeny. The genetically best plus trees were then crossed, and their progenies are now in field trials. Seed orchards will be established based on these trials and used for mass propagation in forest trees. The best tree will be retained and used in controlled crossings, and the cycle will be repeated. Drift and selection, be it artificial or natural, occur at several of these steps. As such, there is a risk of genetic loss. And these are prevented by maintaining large breeding populations, using appropriate crossing schemes, and by conducting selection within families. So keeping genetic variation from all families. In this study, our goal is to measure the change in genetic diversity at some of these steps to investigate whether the risk of genetic loss is adequately averted. And first, we will assess the genetic variation of Scots pine comparing natural and plus trees using markers at the genome scale. And second, we will measure the effects on genetic diversity of selection during the early stages of breeding. So let's talk briefly about the tool we use to obtain genetic data. Scots pine, as other conifers, has a massive and highly repetitive genome. And to overcome these still challenging difficulties in 2022, we have recently designed a SNP genotyping array for Scots pine, the BC50K. So the design of this array went through several steps, from the collection of 4 million markers identified in 8 previous studies, to an in silico evaluation, to the testing of 400,000 SNPs, and to a final selection of the best 47,000 SNPs. This array is great for breeding programs, as it will offer the possibility to use genotype information for breeding. In addition, it's a tool for monitoring genetic diversity in breeding and conservation programs. So the results. So first, let's talk about population structure. We use this tool to genotype about 400 trees from natural populations and about 200 plus trees from 3 areas: southern, central, and northern Finland, which you can see on this map. You can see on the right side the principal component analysis from this data. In green, the natural populations, and in red, blue, and purple, the plus trees. And what you can see is that the natural population cluster with the plus tree from south and central Finland, whereas the northern breeding population diverges slightly. So then we can take a look at genetic diversity. So there are several ways to measure genetic diversity. And here you can see the box plots of the expected heterozygosity in natural and plus tree. You can see that there's basically no difference in expected heterozygosity. But we're going to take a closer look by investigating the variation between the distribution of minor allele frequencies. So to describe first the plot, on the top you see the plot of the count of SNPs at different minor allele frequencies. The distribution starts at 0, which corresponds to alleles fixed in the population. There are two distributions here, one for the natural in dark green population and one for the +3 populations in the lighter shade of green. On the bottom plot, you see the difference between the two distributions. An important precision is that to account for the different sample sizes in the two populations, these distributions have been resampled to an equal size of n equal 80. We see in the +3 compared to the natural tree an increase of the rarest and fixed alleles and the loss of alleles at intermediate allele frequencies. So in other words, a slight decrease of genetic diversity in the +3s. So this could be due to phenotypic selection or sampling. However, it could also be explained by ascertainment bias, which we'll explain now. So ascertainment bias appears because of the way genotyping arrays are designed. More specifically, the design starts with the identification of polymorphic positions in a sample that we can call the discovery population or the reference populations. So here the discovery is consisting 4 individuals represented in the column of this table, and for which we have genotypes at 5 genomic positions, the rows in this table. In variable positions or positions with very rare alleles, here with positions 3 and 4, are excluded of the array, and we retain only the variable positions. So this pre-selection of genetic markers leads to an ascertainment bias, which leads to a distortion of the distribution of allele frequency. So here is an example. You can see the distribution of allele frequencies obtained from samples of Scots pine in Finland. In purple, obtained without ascertainment from exome capture data, and in red with an ascertainment using the genotyping array, the PC50K. And the difference, as you can see, is that we miss a large amount of rare alleles with ascertained data, and we measure an excess of alleles at intermediate allele frequencies. So in our data, the natural and +3s have 2 different ascertainments, so we have to take that into account for our comparisons. And so coming back to the comparison, so to try to get a feel of what the ascertainment bias would, how it would distort these 2 distributions, we run simulations using the same ascertainment as in our real data. And you can see the results of these simulations, the 2, again, the 2 distribution of natural and +3s. And in our simulations, these 2 populations have exactly the same levels of genetic diversity. And you can see that in these simulations, we obtain the same patterns as in the real data. As such, we cannot conclude at this moment about the origin of the difference between natural and +3s in the real data, and we are conducting more analysis and simulations to try to determine whether these differences could be entirely explained by ascertainment bias or not. So as mentioned, we wanted to look next at what would happen during the next steps of breeding. To this end, we compared the +3 and their progeny. And so we used seedlings obtained in controlled crosses of +3s, so 51 progenies of full sibs using crosses from 71 +3s. And we compared the genotypes of offsprings versus the expected genotype based on the parental genotypes. Overall, we see that there is a very so here are the distribution, in red the expected, and in blue the offsprings, and on the bottom the difference. And there is a very weak difference. You have an increase of the rarest alleles and in the offspring compared to the expected, an increase of intermediate allele frequencies. So you have a loss of the very rare alleles. So this again could be due to sampling or selection. So what we wanted also to do was to look at the effect of phenotypic selection. So to this end, we used the seedling data. We simulated an artificial selection within each family picking the highest, the tallest, and the seedlings with the shortest time to bud sets. We retain the top 20% of seedling, and here you can see the difference between the artificially selected seedlings in very light blue against the entirety of the offspring in a darker blue. And there again, you have a small loss of genetic diversity with an increase of the rarest alleles. And so to conclude, this study illustrates the good performance of the BC50K to measure population structure and to gather genetic information in Scots pine. In our comparison of genetic diversity in natural and +3, we observed that there's a small difference of genetic diversity across the genome, but it's essential to account for ascertainment bias, as the difference we observe, even though small, could be actually entirely explained by this effect. However, we note that the diversity at loci underlying selected traits is not specifically examined here, but it will be in future studies. And across the breeding steps, well, overall, we observe little effect of selection of drifts across the breeding steps. However, these effects potentially add up over generations, so it would be important to monitor and simulate the mid to long-term implications. There is a limitation in our analysis. We are using really a subset of the entire crossing data of the +3s. Finally, I would like to acknowledge all the collaborators of this project that have participated in this work, the members of the PUHIRV lab that have provided feedback, and the funding agencies that have supported this work. I would like to thank you for your attention.